Hi students, welcome to SPS syllabus. Today we are going to discuss about, in class 12, we are going to discuss about genetics and evolution. So in the part of genetics and evolution, here we are going to discuss about the principles of inheritance and variation. That is, in unit 6, 7, we are going to discuss about chapter 5. That is, principles of inheritance and variation. So when I come to principles of inheritance, so for this, a person called Mendel has done a lot of contribution to inheritance. So here all living organisms will reproduce and it will form the same kind of offspring. That is, if you see here, a rabbit gives rise to a baby of rabbit. Like the same thing. Lion, like same thing. Okay. Even though it gives us the same kind of offspring, it do not resemble the parent. So it will not resemble the parent. Why? What is the reason? That is, during this process of reproduction, lot of changes will take place. So which will make them doesn't resemble to the parent. Even though they are of same kind, they do not resemble the parent. For example, if you take a rabbit, so a rabbit, even though they are of same of rabbit of the same parent, but they don't resemble. So what is the reason? What happens during the process of? So here, if you take the contribution of Mendel, so when you take Mendel, so originally he is an Austrian monk. So he published, that is, he has done hybridization experiments on pea plant. That is, pea plant belongs to family Fabaceae. Scientific name Pisum sativum. So which belongs to Fabaceae. And his experiments, that is, whatever he has done the research, is published in the form of the translation of the characters, natural of society. So he is the first person to propose the principles of inheritance. That's why he is called father of. So Gregor John Mendel is called father of genetics. The reason is he is the first person to propose, that is, he has proposed the laws of inheritance. Later on, that is after Mendel, again Mendel's laws were rediscovered by three people. That is, one is Hugo de Vries, Poland, Franz Corres in Germany, and Fischer Mark in Austria. So here, these three people has done again the rediscovery of Mendel's independence. So after this, that is, if you see the Mendel's, that is the museum, the place where he has done, that is, this is Mendel University of Brno. This is Mendel Museum, Brno, okay, where he is considered as father of genetics. So before going to the inheritance and variations, you should understand about some terminology in genetics. For example, if you take gene, what is meant by gene? So gene is a functional unit of heredity. Heredity means the transfer of characters from one generation to other generation. So this transfer of characters is responsible by the gene. What is meant by locus? The locus is the position of a gene on the chromosome. So here you are seeing the structure of a chromosome. Okay, the locus is the position of a gene. The position of a gene on the chromosome. What is allele? Allele means the alternative form of a gene which are present on identical locus. That is, so these two are called homologous chromosomes. Homologous means which has, that is, the alternative forms of alleles. So these two are homologous, on which the alternative forms of genes are present on this, that is, which we call it as an allele. So a gene has two alleles. Next one, what is meant by phenotype? So phenotype means external appearance, what you are seeing externally. It may be due to influence of genes or environment as well as factors. And when you come to genotype, genotype is the genetic constitution of an individual, which will reflect the phenotype. Okay. And again, we'll have phenotypic ratio. So the proportion of phenotypes. Genotypic ratio, the proportion of genotypes in it. So like this, we have phenotype, genotype, phenotypic ratio, genotypic ratio. And homozygous. Homozygous means they are similar, that is, the alleles are identical. So here what you are seeing, that is, these are alleles which are identical. Capital T, capital T, small t. These two are called homozygous. Heterozygous means alleles are unidentical in an allelic pair, that is, capital T or unidentical. It's called heterozygous. Like if you write like capital or small, heterozygous. Okay. Capital S, small s, heterozygous. So like this, homozygous and heterozygous. So what is meant by dominant? Dominant means which will express. In a heterozygotic condition which will express, it is called dominant. What is recessive gene? Recessive gene means that fails to express in a heterozygous condition. And next, hybrid. What is meant by hybrid? Hybrid means the progeny obtained by crossing parents, different character. And next, backcross. Backcross means the cross between F1 hybrid, that is the first filial generation, crossing it or not. That is crossing of F1 hybrid with the parents, any one of them. And what is meant by test cross? Test cross is more specific than back cross. Here the test cross is conducted where F1 hybrid is crossed with the recessive parent, that is homozygous recessive. For what purpose will do test cross? To identify the genotype of hybrid. First we look into the Gregor Mendel, what he has done. So when you come to Mendel experiment, so already told, in Mendel experiments, he has taken, for his experiments, he has taken Pisum sativum plant. What is the reason for taking? The reason is, pure varieties are available and it is easy to cultivate Pisum sativum plant. Life cycle of plants is only few months, so that we can get the result early. And many contrasting traits are observed. Contrasting traits means tall, short. Suppose if you take height, a character, tall, short. So flowers are bisexual, where we can do self-pollination. Even the cross-pollination, they can be done manually. Hybrids or fertile. So these are the reasons why Mendel has selected pea plant as an experimental. So here when you come to the pea plant, so here what you are seeing, he has taken 7 pairs of contrasting tribes. That is, if you take the character height, so in this the contrasting tribe is tall. We come to the shape of the seed, the character shape of the seed, round, wrinkled, contrasting tribes. Seed color, yellow, green, the contrasting tribes. Seed coat, that is the color of seed coat, green, white. Pod shape, inflated, full, no restriction, constricted. Pod color, green, yellow. Flower position, axial, terminal. So these are the contrasting traits, and these are the characters which are taken for Mendel for his experiment. In the contrasting traits, there is a dominant trait as well as there is a recessive. So dominant means which will express in heterozygous condition. Recessive means which will fail to express in a heterozygous condition. So what he has done? On taking this Pisum sativum, so Mendel has proposed 3 laws, that is, law of segregation, law of dominance, law of independence. So he has proposed, Mendel has proposed 3 laws, law of segregation, law of dominance, law of independence. So what is this? We'll see. So here when you come to law of segregation, what it says, what is meant by segregation? Segregation means separation. So during the formation of gametes, alleles are separated. For example, if you take, this is an gene with alleles capital Y and small. So during the formation of gamete, that is gametogenesis, capital Y goes to one gamete, that is gamete with capital Y, gamete small y. Here also the same, that is here the alleles are separated. The gametes are getting only one of the allele gene. So law of segregation, what it says is the alleles are separated during the formation and the fertilization takes place randomly. First we'll start with what the experiment, what he has done. So in his experiment, he has taken 2 parent plants. That is, he has taken a pure tall plant, that is homozygous tall plant, pure dwarf. So homozygous tall plant means the alleles are capital T, capital T. Whereas pure dwarf, the alleles are small. So here we will get, the gametes will get only one allele, that is capital T, this is small. So this is next step is fertilization, where fertilization leads to formation of F1 hybrid. This is F1 hybrid, which is heterozygous tall, that is capital T. F1 hybrid is heterozygous tall, which is tall heterozygous. So this F1 hybrid is subjected to self-pollination. When it is done self-pollination, both the parents are heterozygous tall. Then here we will get 2 types of gametes, one with capital T, one with. So when they are crossed with, we will get 4 offspring. Total we are getting 4 offsprings in F2 generation. In this, one is pure tall, 2 are heterozygous tall, one is homozygous. That is, if you take the phenotypic ratio, the ratio is 3:1, number of tall plants to number of dwarf plants. When I take genotypic ratio, that is the pure tall plants, homozygous, capital 1, heterozygous 2, and homozygous dwarf, that is the genotypic ratio is 1:2:2. So like this, in a monohybrid cross, he has taken only one character into consideration and started crossing. So in the process of crossing, we use, that is, Punnett space. What is this Punnett space? What is the use? This Punnett space or device, that is by Reginald C. Punnett. So that is, this Punnett square is used to predict the genotypes of a particular cross. In a breeding experiments, to predict the particular genotypes of particular cross are used. So here you can identify the predict the genotypes, as well as we can probability of an offspring of a genotype can be detected. So here you can see the best example, which is given F2 the generation when their F1 hybrid is self-forming. So this is the gametes of one parent. So these are gametes of one parent. That is, for example, if you take the female parent, the male parent. Okay. So these are gametes. So here when you take a Punnett sphere, when it is crossed, so here you can see the crossing, these two will get capital T, this capital T. So a Punnett square can be used to predict the genotypes of a cross breeding experiment, as well as to determine the probability of an offspring. So here this is the overall the Mendel cross which he has done. So here he has taken homozygous tall plant. Homozygous. While you are writing any cross, you should start with the parents. P means parents. So in the parents, that is, first you should write the phenotype. Tall. Genotype, homozygous dominant, that is homozygous tall, homozygous. When you write the gametes, next is gametes. When you write the gametes, here we will get capital T, here we will get. So fertilization, it leads to capital T, that is heterozygous tall. So this is F1 hybrid. And in this F1 hybrid, it is self-pollinated. Then you will get here types of gametes. One gamete is the capital T, one gamete. Totally you are getting 4 offsprings in F2 generation. The phenotypic ratio is 3:1 and 2:2. So like this, Mendel has done a monohybrid cross. So whenever you are writing a cross, this is the procedure. You should start with parents, prototype, genotype, gametes, fertilization, F1 hybrid. Again, the next F1 hybrid, self-pollinated, F2 generation, the pair with phenotypic and genotypic. So based on the law, that is based on monohybrid cross, Mendel has proposed the law, that is law of dominance. What is this law of dominance says? The law of dominance says is every gene has 2 alleles. Whatever you take, the gene has 2. For example, if you take, that is, if you take here capital R, is a is 2 alleles. The same like any gene, if you take. In this, one is dominant, one is recessive. Dominant means which will express. Dominant, recessive means which fails to express. So here, what law of dominance says is, in a gene there will be one allele will be dominant, one allele will be recessive. Dominant means which will express. Allele means which fails to, due to the presence of dominance. So that is the reason why heterozygous condition will always be showing dominance. So here you can see this is red and white flower or small crossway. So it is a red. The same thing you can apply to tall also. That is, when you take tall and a dwarf plant, the gametes will these basically you get an heterozygous tall. So based on the monohybrid cross, he has proposed, that is the law of dominance. And in this monohybrid cross, for example, if you don't know the genotype of F1, unknown genotype, that is F1 hybrid is unknown genotype. So to find out the genotype of F1 hybrid, will perform test cross. So test cross is crossing F1 hybrid with recessive pairing. Here F1 hybrid is capital T. This recessive parent is. Here you will get two types of gametes, one with capital T, one with small. Here only one type. So when they are crossed, you will get one heterozygous tall, one dwarf. That is, the phenotypic ratio is 1:1 and genotypic ratio is. So what it will indicate? When the ratio is 1:1, it will indicate that the F1 hybrid is heterozygous. When F1 hybrid is heterozygous, we will get the phenotypic ratio, genotypic ratio, test cross will be 1:1. Suppose if it is homozygous, for example, if it is homozygous, the F2, the phenotypic ratio will be 1 and as well as the genotypic ratio. So, in order to identify the genotype of F1 hybrid, we will perform test cross. So here you can see the examples of a test cross here. Okay. So here they have taken, that is the genotype gametes from parent of unknown genotype. So here if you see this unknown genotype, we don't know the genotype. Here we have done the crossing. A test cross is resulting all dominant offspring. All the offsprings are dominant. What it indicates, the F1 hybrid is. If it is resulting in 1:1 ratio, 1:1 ratio means the F1 hybrid is heterozygous. F1 hybrid is heterozygous. Here, the F1 hybrid is homozygous. That means if we get ratio 1, it indicates homozygous F1 hybrid. If you get ratio 1:1, it indicates the heterozygous, that is F1 hybrid is heterozygous. So this is the ratio of monohybrid test cross. So what is meant by test cross? What is meant by back cross? So test cross is the breeding of F1 hybrid with recessive parent. But back cross means it is a crossing of F1 hybrid with one of the parents. It is not mentioning specifically whether specifically dominant or recessive. So all test crosses or back crosses. In back cross, that is, if it is crossed specifically with recessive phenotype, it is test cross. That means all back crosses are not test. All test crosses are back crosses, but all back crosses are not test cross. So this is the difference between a test cross and a back cross. Here you can see the F1 hybrid is crossed with recessive genotype, but here it is crossed with either homozygous or heterozygous. Okay. So test cross will identify the genotype of the hybrid. Back cross will recover the genotype. So till now, what we discussed is, that is about the monohybrid cross and the law of Mendel, which is proposed, means law of segregation. So based on monohybrid cross, Mendel has proposed, that is the law of dominance and law of segregation. When you come to law of dominance, what law of dominance says is, that is in a gene, that is if you take in a genotype, there will be a dominant allele. There is a dominant means which will express, the allele means which is not able to. So here there are few deviations from law of dominance. The best example for this is you can take a plant called Mirabilis jalapa, in which a deviation is observed by coherence that is incomplete. What is this incomplete? Incomplete dominance means the dominant allele is not able to show complete dominance. So then what will happen if it doesn't show the dominance? Instead of getting any parent genotype, we'll get an intermediate. So here we'll see with an example. That is, if you come to Mirabilis jalapa, in this there are red color flowers as well as white color. So when you take the crossing, you should get that is a red color flower. That means according to Mendel, we should get the F1 hybrid should be a red color. But here, interestingly, what we got is a pink color. Means neither red nor white color. What we got here is the pink colored flower. So here if you see this, here if you take this, that is the red colored flower. When it is crossed with white colored flower, the gametes will be, here you should get capital or small or, that is F1 hybrid. According to Mendel, this should be red. Experimental report is the pink flower. So why here we got pink instead of red? According to Mendel, you should get red. Here we got. What is the reason behind? The reason behind this is the dominant allele which is present. The dominant allele means here capital O. The recessive allele means the small o. It is not able to show the complete dominance over the small o. Actually, according to Mendel, the dominant allele will show the complete dominance. But here it is what is happening is it is not able to show its dominance. That's why we have pink color. That is the reason why we go to pink color. Okay. We will follow the Mendel cross. What he has done in Mendel is what he has done in the mono. So now we got F1 hybrid. So this F1 hybrid, that is self-pollinated. Hybrid is self-pollinated. Self-pollinated means capital or capital or small. So what we will get here? We will get two types of. Here also types of. So by the end of the result, that is if you take, we will get two red flowers. That is by the end of the result, we will get red flowered plants will be 1, pink colored plants will be 2, and white colored plants. That means if you take the phenotypic ratio and genotypic ratio, both will be 1:1. So this is for a deviation from Mendel's law. According to Mendel's law, the phenotypic ratio should be 3:1. But here we are getting, so which is a deviation from Mendel's law. That is, by the end of F2 generation, 1 red flower, 2 pink flowers, 1 white flower, where the phenotypic ratio or genotypic ratio is 1:1. Same, instead of 3:1, here we are getting 1:1. And this concept is called incomplete. And next we will come to another deviation, that is co-dominance. What is this co-dominance? Here co means both. So in this co-dominance, what happens is These two codominance, both alleles in a gene will express. Generally, only one dominant allele will express. But here, both the alleles will express. That means the genes that are present in allele will express, which result in. So here we'll see with an example. The best example for codominance is, if you see the blood groups, we follow generally ABO blood group. Here we have A blood group, B blood group, AB blood group. And these blood groups are controlled by three alleles, IA, IB, I. So when you take A blood group, it has a homozygous genotype and a heterozygous. Homozygous is IA, IA. Heterozygous is IA small. B is also homozygous genotype and it shows heterozygous genotype. IB, IB, IB. But when you come to AB, only heterozygous IA, IB. And O both small. So here we can see codominance in the AB blood group very clear. In AB blood group, the genotype is IA, IB, where in this any one should express, but here both are expressing. When both will express, instead of getting A or B blood group, B blood group. And when you come to blood group is not only example for codominance, as well as it is an example for multiple. What is meant by multiple alleles? So generally a gene, that is if you take any character, there will be alleles. Whatever you take, if there are more than two alleles, then it is called multiple alleles. That means if you take the blood group, it has three alleles, IA, IB, I. So blood groups is controlled by three alleles, IA, IB. So here you should remember the phenotype of blood group as well as the genotype of the blood group, where we'll come across crossings in the competitive exam. That is, in the NEET examination, they may ask crossing. That is heterozygous A blood group crossing with B blood group. So here it is, you can see. Here IA and IB are codominant, whereas I small I is recessive. So which is a recessive allele. So here if you take A blood group heterozygous, IA will express, small I will not. The same IB express. So these are the genotypes in the blood groups A, B, AB. Here you can see what are the possibilities of having blood groups when the parents are of a particular blood group. Possibility of the children have. Suppose if A blood group is there for both parents, the possibility is there will be A as well as first. The points answer. If AB or their parent, one parent is A, another parent is B, all the types of blood. If A blood group and AB blood group, A, B and O, A or O. B and B, B or O. Take B and AB, A, B, AB. B and O, B or O. AB and AB, again A, B, no. AB and O, A or B. O group, O group. So these are the probability of getting the children when the parents are having a particular blood group. So this blood groups which are present is an example for, the blood group is an example for both, that is the codominance as well as, which is a deviation to the Mendel's laws. According to Mendel, a gene will have two alleles, that is a character is controlled by two alleles, and according to Mendel, is dominant allele is completely dominant over recessive. But in incomplete dominance, it is quite reverse. We come to next. So generally, a gene will control a character, but here in pleiotropy, when it comes to a single gene will control many phenotypic effects. Like, for example, if you take the blood cell, that is red blood cell, which is disc shape. But when the red blood cell in a genetic disorder, when it becomes into a sickle shape due to a genetic disorder, it is called sickle cell anemia. So when this sickle cell anemia occurs, it will show that is many symptoms, multiple symptoms. That means the gene which controls the shape of this will control many other characters also. So this is a sickle cell anemic patient, the red blood cell shape, and a normal blood cell will be like this. So this is pleiotropy. Pleiotropy means a single gene have multiple phenotypic effects. Here you can see. So sickle cell anemia, which is due to a gene mutation, will affect that is blindness, liver failure, heart attack. Like this, multiple traits. So one single gene will show many effects. That is many phenotypes. In a sickle cell anemia, due to this, it leads to blindness, liver failure, heart attack, all. So by the end of that is the monohybrid cross, we have understood that the law of dominance, law of segregation, and their deviation. That is the deviation that is incomplete dominance. The next one is co-dominance, multiple alleles. So now we are going to discuss about another experiment which is conducted. That is the last law, which is law of independent assortment. Assortment means separation. So here in law of segregation already we studied that alleles separate during formation of, and this alleles separate independently. So the separation of alleles doesn't depend on other type, that is other. So this law is proposed after performing dihybrid. So what is meant by dihybrid? Here he has taken two characters into consideration. In monohybrid, only one character is taken into consideration. Here, two characters. So here he has taken seed shape and seed. That is, when you come to color, yellow is dominant, green is round shape is dominant, that is wrinkle is recessive allele, round is dominant allele. Yellow is dominant allele, green is recessive. So this law of independent assortment is proposed, that is, he has proposed this law of independent assortment. So in a dihybrid cross, when you start with dihybrid cross, so first we'll start with, he has taken pure, that is homozygous round yellow parent crossed with homozygous wrinkled green. So capital R, capital R, capital Y, that is round yellow, homozygous round yellow parent is crossed with homozygous green, is wrinkled green. So when they are crossed, so we will get, that is when you take the gametes here, we will get capital R's here. After fertilization, we will get, that is capital R, that is heterozygous round yellow, that is F1 hybrid is heterozygous yellow. So this F1 hybrid is self-pollinated. The same procedure, it has pollinated, self-pollinated. And it is self-pollinated. The parents both will be, that is capital R, small r, capital Y, small y. Here also capital R, small. So in this parent, from this parent, we'll get 4 types of gametes. That is capital R, small y, capital R, capital Y. Small r, capital Y, small r, small. The same type of gametes we'll get here on the other side. That is capital R, capital Y, capital R, small y, small r, capital Y, small. And when these gametes are crossed by using, that is, a Punnett scale, they are crossed by Punnett scale. So this Punnett scale, if you see, that is, we got, that is, by the end of this, and that is in F2 generation, there are 9 round yellow there are 9 round yellow three round wrinkled, 3 are round wrinkled. These 3. Next, wrinkled yellow. These 3 are the only one that is left out is, that is, wrinkled green. So that means total we got 16 offsprings in F2 generation. This 16, 9 are round yellow, 3 are round green, 3 are wrinkled yellow, 1 is. That is, by the end we got the ratio is 9:2. That is, the phenotypic ratio is 9:2. So by the end of dihybrid cross, Mendel has got the ratio that is 9:2. So here you will see, after the end of dihybrid cross, if we perform the analysis, that is 9 out of 16 yellow, 3 out of 16, that is round, 3 out of 16 wrinkled, 1 out of 16 is, that is completely. Then we got so after this analysis of dihybrid ratio. So here if you see this dihybrid cross, here a green pod, that is, if you take a green colored fruit, that a green pod can have a wrinkled seed or round seed. At the same time, an yellow pod can have a round or wrinkled. Same thing applies to yellow as well as wrinkled. That means the different traits do not influence the inheritance of each other. That means when you take the F1 hybrid, so during the formation of gametes, the alleles are separated independently. That means capital R may combine with Y as well as combine with small y. The same thing applies to small r. Small r may combine with capital Y, or that means the alleles are separated independently. They are separated. So this separation of alleles independently during the formation of gametes, it is called, that is called law of independence. So based on dihybrid cross, Mendel has proposed law of. So according to law of independent assortment, alleles are separated independently during the formation of gametes. So this is the final test which we got the final result. The phenotypic ratio, that is when you got the final. And in dihybrid cross also we can perform the test cross. To find out the genotype of F1 hybrid, we can find out the test cross. So here in test cross, if the F1 hybrid is heterozygous, that is if the F1 hybrid is heterozygous, in test cross we'll get the ratio 1:5. If it is homozygous, we'll get only 1. That means if the test cross ratio is 1:1:1, it is only, that is the F1 hybrid is heterozygous. So it shows the F1 hybrid is heterozygous. Suppose if you got 1, only it shows the F1 hybrid is homozygous. So this is the ratio of dihybrid test. In this also, later on there are few deviations like polygenic inheritance is also called quantitative. What is meant by poly? Poly means so one gene, one character is controlled by many. For example, one, according to Mendel, that is if you take one character is controlled by one gene. But here there is a deviation. One character is controlled by many. That is many. For example, if you take the human skin color and eye color, height, intelligence, the kernel color of wheat. So these all are poly. Poly means one character is controlled by many genes. For example, if you take human skin. The human skin is controlled by 3 genes. That is gene 1, gene 2. The people who are having very light color, they are having recessive, all the recessive. The humans which are, that is the dark one, that is all or tom. So that is when you write the gametes, here we'll get capital A, B. Here when you write the gametes, we'll get that is capital A, capital B, capital C, here small a, small c. So when you cross these two, we'll get the F1 hybrid will be capital A, small a, capital B. It is an medium color. That is, the dark color is influenced by, that is, all dominant alleles. The light color is influenced by all recessive alleles. In between these, what we get, the intermediate, that is the medium one, which has the mixed combination of dominant and recessive alleles. Here you can see, that is, if you cross, that is, if you perform the same like Mendel has conducted the cross. So here you can see, that is, the complete dominant, the darkest one, or this. So the lightest one, if you see the graph like this. So here the graph shows the number of individuals, the of darkest, the number of individuals, medium, that is, as we move, that is, from dominant to from dominant alleles to gradually when we move to recessive alleles, the color is becoming gradually. So these all are medium color. This is dark, this is white, light. Remaining all are medium in color. So until the, that is when you come to Mendel, during the time of Mendel, there is no scientific awareness. Okay. That is when you come to the Mendel's the experiments. So the Mendel's experiments were came to light after a long time of his proposal. That is during the time of Mendel. So there is no communication. There is no communication. There is no proper communication. And Mendel, while during his experiments, he has proposed, that is, he has used many mathematical logics in consideration. The scientific society is not able to understand. So there are few reasons why Mendel's experiment, that is, Mendel's experiments were not determined or determined after a very long time. The reason is lack of communication, and he has used mathematical logics, many statistical aspects he has used to find out the ratios. He has used many statistical principles. The scientific society is not able to understand his. He has taken many number of offsprings into consideration. Like this, he has where the scientific society is not able to. Later on, Correns, Teschermark, Hugo de Vries, they rediscovered the Mendel's law. Hugo de Vries, Correns, Teschermark has rediscovered the Mendel's law. After this, Correns, Teschermark, and Hugo de Vries. Later on, after this, there is a lot of advancement in the field of biology. Their genes, chromosomes were discovered. Okay. And the cell division meiosis was studied well. Many things are revealed. So based on the Mendel's theory, so the chromosomal theory of inheritance is, what is this chromosomal theory? It was proposed by Walter Sutton and Theodore Boveri. It is proposed by Sutton. So they worked out, that is, they have found out chromosome movements during, and they identified the chromosomes, the movement of chromosomes was parallel to the behavior of the gene. So they use this chromosome movement alignment. That is, chromosomal theory of inheritance is an combination of Mendel's laws with chromosomes. So here, the knowledge of chromosomal segregation with integrated Mendel's principles, it is called chromosomal. According to this, chromosomes and genes are present in pairs in a diploid cell. That means in a diploid cell, if you take, chromosomes are present in, that is, you can write in like this 23 pairs of first pair, second pair, third pair. During the formation of gamete, that is meiosis, homologous chromosomes separate. During meiosis, what happens is the diploid number becomes haploid. That means gametes are always haploid. And due to fertilization, the restoration of diploid number takes place. That means in fertilization, gametes, haploid gametes form diploids. That is, in chromosomal theory of inheritance, Sutton and Boveri studied the chromosome movement as well as to explain the Mendel's law. Later on, after Mendel, a person called Thomas Hunt was born. So he has conducted experimental verification of chromosomal theory, and he is called Thomas Hunt Morgan, is called father of experimental genetics. So here he has conducted his experiments on fruit fly. Fruit fly scientific name is Drosophila melanogaster. So which is a dioecious. Dioecious means where the male and female fruit flies are present. So here what you are seeing is a female fruit fly. This is a dioecious. It is sexual. And these fruit flies has many advantages. Why he has selected only fruit fly for his experiment? Why he has not taken The reason is they can be grown on simple medium elaborate, and their life cycle is about just only 2. That means we can get the result early within 2 weeks. And in a single crossing, many progeny will be produced. And there is a clear differentiation. We can identify phenotypically, we can differentiate the male and the female fruit. And we can see many variations within low power microscope. So if you see the male and female, the abdomen of female is broad, whereas the male is narrow. So like this, we can differentiate the male and female fruit fly easily externally. The life cycle is only just 2 weeks. When it is 2 weeks, the result we can expect is early result, and they can be grown easily in the laboratory. And we can perform the mating, and we can study the hereditary variations. So here, this is the reason why Mendel has selected the Drosophila. Here you can see the differences between the male and female fruit flies. If you take the male, is short, round abdomen, black spot, bigger black spot on the. Here it is broad abdomen. And female is larger when compared with male. Female is larger, and so we can see clear difference between and female. So this has led, these are the features which has led the Thomas Hunt Morgan to perform experiments on the female, that is the Drosophila melanogaster, which is commonly called fruit fly. Okay. So till now, we discussed about the Mendel's contribution to genetics, the monohybrid cross, the dihybrid cross, and the next one is we discussed about the deviations from the dihybrid as well as the monohybrid cross. Along with, we discussed the 3 laws of Mendel's, that is law of dominance, law of segregation, law of independence assortment. So follow us on social media. Thank you very much. We'll meet in the next session.